is using that to fuel carbon reactions. What else does it use other than ATP to do the synthesis part? CO2? Anything else?
Water.
Water is used in the light reaction. NADPH, yes. NADPH, and yes, the rubisco is the enzymes in the steps. Since we were talking about stomata, I forgot to show you water lily. So you know how we said, so what did I say? Actually, I'm going to ask you guys, what did I say where the stomata are found? Are they on underside or are they on top?
Under.
Under, right? So for water lily, you see the underside of the leaf is in water. So it cannot really get any gases through there. So for water lilies, stomata are going to be on top. So that's a modification. So we went through the photosynthesis and we said it's going to make ATP, NADPH, and then it's going to use that to make glucose. Now we have glucose. Where's that glucose going to go? It's going to go make the cellulose. You guys remember monomers and polymers? It's going to make long chains of cellulose. The cellulose is something we cannot digest. That's why I said people can eat salad all day long and not gain any weight because your body doesn't digest it. Bacteria breaks it down. So the cellulose is going to give it structure. And other thing that it can store it is as a starch. Starch, we eat this. So plants, they make it for themselves, but we started eating these. So we take this starch. That's also a polymer. I showed you water lily. That's one modification. That's how it can get gases. And we learned that through stomata, carbon dioxide can go in, oxygen can come out, and also water can come out. So if the stomata remains open all the time, it's going to lose water, especially if you think about the desert. There's still plants there, but there is not as much water. So if the plants, they had stomata open maybe during the day there, then they will lose a lot of water. So we're going to look at some modification or different type of plants. There's C3 plant and C4 plant. I want you to look at these both images and tell me what's different. Is there anything that you notice? Shape. Be a little bit more specific and look at maybe the cells, maybe the size of the cells. So if you were to take an orchid and put it in the desert, it's not going to survive because it has the stomata on the top. It's going to lose a lot of water. Vascular tissue is bigger. So this has this something called bundle sheath cells. That's the vascular tissue. And it's so there's I don't want you to worry about the veins. I just want you to know C4 plants compared to C3 plants have bigger bundle sheath cells. And the reason for that is that it goes through, it doesn't use rubisco. It does use rubisco, but at a later stage. It goes through a different enzyme and different steps. It's a little bit longer steps that it uses to get glucose. So all I want you to know, it has a bundle sheath cells that's going to allow it to basically survive when there's not a lot of water available. And then there are camp plants. These are the desert plants. So notice the different leaf anatomy in the Calvin cycle. So the Calvin cycle happens in the bundle sheath cells here, part of it. So it happens a little bit differently compared to here. That's all I want you to know. Then we have camp plants in the desert. These don't have their stomata open during the day. They have their stomata open at night. So they have their stomata open at night. At night, they take the carbon dioxide, they store it, and they use that during the day to make that, do the light reaction. Does that make sense? So they're storing that carbon dioxide. They're being smart about it. If they left it open during the day, then they would lose a lot of water. They wouldn't be able to live. So that's evolutionary adaptation that they did. Now the stomata is underside. Sorry, not underside. They open at night, and they take in carbon dioxide, and they use that carbon dioxide during the day to do the light reaction. So they don't lose water during the day. Here, like sugarcane plants, they have a different pathway because of those bundle sheet cells. So it happens twice. It uses, I think, PEP enzyme. You don't have to know the name. Then it uses rubisco. And this is what the normal pathway C3 plant. So those are different kinds of plants. And they have different types of environments. Like I said, orchid can't live in this environment. Only these type of plants can live in these environments. So limitation. So this photosynthesis part, it's called photorespiration. This is ATP cost because it's a longer pathway. It's going to take a little bit more energy to make that glucose. And this here, reduced carbon availability, it can only use as much carbon that's being stored. It can't really open the stomata to get more carbon during the day if it runs out. Then it just have to wait until the night and store more carbon. So it has reduced carbon availability. Here we have bundle sheet cells. Light reaction and carbon reactions occur in separate cells, and that's the modification of bundle sheet cells. And then for CAM plants, CO2 is observed at night. Light reaction and carbon reactions occur during the day. The environment is cool and moist for this, hot and dry for these. Any questions so far? You guys are good? Let me see what this video is. So I'm gonna try to draw it out using this screen. So let's say you have a plant cell. This is a plant cell here. And I'm talking about when you cut out a section, maybe on the plant cell. And then you have inside, you have these organelles. What this organelle is a chloroplast. Stop me when it doesn't make sense, okay? So what I'm gonna do, I'm gonna try to zoom in on this chloroplast. So let's zoom in on this chloroplast. So now this is what you have. This is a chloroplast zoomed in. You have two membranes. And inside this, you guys remember what this is called? Which membrane is this and which one is this? So this is called outer. So what's the other one? What's the other one called if this is outer?
Inner.
Inner, right? Inner membrane. I apologize for messy letters. It's really difficult to write using this. And then inside, you're gonna see these stacks. What do you call these as a whole?
Granum.
Granum. If you're talking about all of them, it's called grana. Now, if you were to just focus on this part of the granum, or sorry, actually, what is this single one called?
Is it the thylakoid?
Thylakoid. Now, in that thylakoid, Let's see, be green color. What do you call this part of the thylakoid? The highlighted part in green.
Is it the pigmented molecules?
It's the thylakoid membrane.
Oh, membrane.
Now there's this space, the white space. I'm gonna try to use different color to highlight that. So this white space, or this white space, what is that called? Again, it's this thylakoid. This is thylakoid membrane. So this space is gonna be called thylakoid space. There's this fluid. There's this fluid that's found in here. You guys remember what that is called?
Stroma.
Stroma. So I'm gonna just put fluid in brackets. And that's called stroma. So what I'm gonna do next, I'm going to zoom in on this thylakoid membrane. So let's zoom in on this thylakoid membrane. So I'm gonna make a box here. That's what I'm gonna use. So this is I'm gonna try to draw it inside this box. So this thylakoid membrane, I zoomed in on it. So you have this membrane here. What is this made of? You guys remember? What is this thylakoid membrane made of?
Phospholipid bilayer.
Phospholipid bilayers. So you have, I'm not gonna, complete it, but this is just to give you an idea. It's made of phospholipid bilayer. So this is the thylakoid membrane. If this is the membrane, what's here?
The thylakoid space.
Thylakoid space. So this, or also known as lumen. And what about here? What's here? In this area. So we're looking at this. If I'm pointing here, then I'm talking about this area.
Is that the stroma?
That's the stroma. Now, what's found on this thylakoid membrane? You guys remember? What's found on this thylakoid membrane? We said it has a larger surface area, so it can have more molecules. Those molecules are those protein channels, you can say, or the parts where the electrons get excited, so the photosystem. Okay, so we have here, there are a lot of them. I'm not gonna try to draw all of them. I'm just gonna try to draw what we covered so far, okay? So I'm gonna draw one here. What is the first one called? This photosystem, which photosystem comes first? Photosystem 1 or photosystem 2?
2.
2. So I'm gonna say PS2, not PlayStation, but photosystem. So photosystem 2 comes first, and then you have photosystem 1, and that comes second. So I'm gonna draw another one here. You have photosystem 1 here. In between, I use different color here. In between, there are other, F6 cytochrome, a lot of others. So I'm just gonna draw those. You don't have to know the names. I just want you to know that it's not only these two. There are multiple. There are multiple there. So this is the light part of the photosynthesis we're talking about. So this is the light part. I don't know why it's still writing yellow. So this is the light reaction, light dependent reaction, or where it's going to use light. So what's going to happen, photon, sunlight, falls down and it excites this electron in the chlorophyll A system, or the molecule chlorophyll A. So this is a photon. It's going to come down. It's going to excite the electron. So this electron gets excited and it goes through this photosystem 2 and it moves down the electron transport chain from maybe this channel to this channel to this channel. And as it gets excited, so I want you to write this down. I'm not going to be able to type this much. So when it gets excited, it goes in high energy state. So when it gets excited, it goes in high energy state. Then it starts to go through these molecules and it starts to go down in energy state. By the time it reaches photosystem 1, again, it's low in energy state. Another photon hits here, light energy. It gets excited again and it goes through the molecules again. And the last molecule that goes to, so I'm going to try to draw it here, is the NADPH plus. That's the final electron acceptor that turns into NADPH. I'm trying to write NADPH here. I don't have space. Please write that down on your own. So where is this electron coming from? Where is this electron coming from?
Is it coming from the H2O molecule?
It's coming from the H2O molecule. So you have H2O molecules that's going to give the electron away, and this is going to be turned into molecular oxygen. And then the electrons from this are taken, and those are the ones that are getting excited. So I want you to say that oxidizing water, oxidation means losing electrons. Oxidizing water is going to convert it to molecular oxygen, and it's going to give the electron to this photosystem. And then NADP plus is the final electron acceptor. There's one more thing I have left out so far. So what did I tell you guys as it moves from high energy state to low energy state? What is it doing? Where is that energy going? There's one more part that's missing still. Now think about ATP synthase maybe.
Is the energy going to the reaction center?
Energy is being used to take these hydrogen protons or hydrogen ions to pump them inside in the thylakoid space. So you have high concentration of hydrogen ions inside compared to the outside. And this is when I said it's like a balloon. is filling it up with hydrogen ions. So it's putting more and more hydrogen ions inside in the thylakoid space. This is the thylakoid space. And these hydrogens, because they're in high concentration here and low here, they want to leave. They want to move down the gradient. The way they're going to leave is through ATP synthase, and I don't have a space to draw that. So I kind of ran out of space. Should have done it here. But the ATP synthase is also found on the membrane, but it's using these hydrogen ions when they leave to put ADP and phosphate together. So that's going to give you ATP. So that's another maybe protein engine, you can say, and through this hydrogen ions leave. And as that happens, this is what you get. So I know I drew it here. It's supposed to be on the membrane here, but that's what's happening. So did that help a little bit? Yes? You guys understand it a little bit better now?
Yes.
Okay. Any questions? I'm gonna type the final basically products.
Do we have lab today?
No lab today. I sent it out last week. Not last week, Monday. I sent out the recording. It was just a review. It wasn't really a lab. No lab next week either. It's a short week. I'm trying to find where I can type it.
Professor, when it goes from NADP plus to NADPH, is it just attaching a hydrogen bond, or?
It's giving it the electron to NADPH. So it's the NADPH is the final electron acceptor. It accepts the electron. It's like Uber,
Okay.
like I said. So what's going in? So water is giving electrons. So this replaces electrons. And this is light reaction we're talking about. So what I just went over, I haven't covered the dark reaction. Okay. So water replaces the electrons, and those electrons pump the hydrogen ions inside, and it's going to use ATP synthase to produce ATP. And another thing is the NADPH. That's the final electron acceptor. And it's going to accept the final electron. So that's what's happening in the light reaction. You're getting ATP and you're getting NADPH. That's what's going to be used in dark reaction along with CO2, and then oxygen is a byproduct. In dark reaction, you're going to use, for this I said you need to know what goes in and what comes out. Use CO2, and then Rubisco is going to basically attach those CO2 molecules together and produce glucose, which is a 6-carbon sugar. And then it rearranges byproducts to make more Rubisco. I'm gonna say RUBP. Any questions? No questions? Okay, so no questions.
I had a question.
Yeah.
Is there anywhere we can take like a practice? I'm not really good with exams for like this kind of stuff. Is there anywhere we can take like a practice or
So your homework on McGraw-Hill serves as a practice. So the question on the exams are similar to your homework questions.
Okay, so just study those really good and. it should-
Yeah, so make
sounds
sure
like
you're
okay.
doing the homework. So that gives you the practice for the exam.
Okay, thank you.
Professor, when is the